Next presenting is going to be Flomics, Joao Curado, CEO and founder of the company.
Okay, good afternoon. I'm João, and I'm here to tell you about Flomics, fluid transcriptomics. The problem is quite clear. I'm sure everybody in this room suffers from it directly or indirectly, but there's a lack of non-invasive early diagnosis on most of the most deadly diseases in the world. We are still using procedures that were invented more than 50 years ago, and these are very time-consuming, they are very costly, and more importantly, they produce errors. That's why we are bringing Flomics liquid biopsies with RNA molecules. So from a standard clinical blood sample extracted into a special tube, we can analyze the molecules of RNA and extract the same information that doctors nowadays obtain when they perform a standard biopsy, which is of course very invasive because they need to open the patient and extract the diseased cells. The magic behind the method is that we are looking at RNA molecules. RNA molecules are DNA in action. They are the first manifestation of the cells when they face an external stimuli or threat like a disease. This is a novel technology, an untapped source of biomarkers. And it's such a dynamic molecule that the usage is very broad, and it's not only restricted to DNA mutation, to mutation-driven diseases such as DNA, for example. We can use it for diagnosis to patient stratification. Our first market entry, our first test is in colorectal cancer. This is the second deadliest cancer in the world, mainly because we detect it too late. When we detect this cancer, it's already too late to perform surgical intervention, and the chances of survival are reduced to half. We partnered together with a hospital in Santiago to develop Flomics Colonizer. From a standard blood test, as I told you, we detect the disease in less than 3 days and with 100% accuracy. There are many competitors in the field we are aware of, but most of them use stool-based analysis, which the patients don't like, so they are not very fit to be used as screening, and they don't have the same sensitivity or multidimensionality that the dynamic RNA gives us. So we can use it for prognosis, to patient monitoring, and even in the future, as we accumulate data, to predict the best treatment for each case. In order to reach the market, we are planning to raise 3 million from public and private funding in the next 3 years, and with the 10K from the Global Accelerator, we plan to hire the best experts that can help us in the definition of the IP strategy and the regulatory and clinical plan that will help us to reach the market in 2022. We are a truly multidisciplinary team. We are all scientists, so we came from a scientific background, but we diverged very early in our career. I'm specialized in bioinformatics and machine learning, Esther in molecular biology, Nuno in business development and startup growth, and Luis Corrodi in regulatory and product development. We also have strong advisors in our board, from science to the clinical partners and the doctors in the hospitals, as I told you. So thank you very much for listening, and I hope to hear your questions and feedback.
Thanks, Joao, for the very nice speech. So his question?
Do you have some KOL supporting?
Yes, so here in the field of colorectal cancer, Ceres Fernandez comes from the Santiago hospital, so they are experts in the field, in the clinical diagnosis of colorectal cancer. Rodrigo is a KOL in RNA, which is our main technology that we have behind.
So, but in colorectal cancer, you have Dr.
Ceres Fernandez and her team, yes, they are helping us not only with the samples that they provide us, but also with the knowledge and to really know what is the best way or where is the right point to perform the analysis, how early we should try to diagnose and this type of things. We trust them because we don't have medical education.
Okay, thank you.
Will you pick with this analysis very early stage, the kind of the one that are contained in the polyp and are not still invasive, nothing like that, like stage 1 or below? Because you need functionality and you need significant activity to release the signals on the bloodstream, and usually those localized tumors, which are the ones that are amenable to surgery, which are the ones that you want to target first, are not detected unless you take out the polyp and look at the biopsy of it, right?
So we have samples from stage 2 to stage 4, and in all of them we can detect the disease. And actually we are detecting genes, so our RNAs that we detect as part of our biomarker model, they were previously identified in the literature as cancer drivers, but from normal biopsies. So from actually cancer cells that were extracted, they detected these biomarkers that we are also detecting circulating in plasma, which gives us some expectation or trust that these are actually RNAs that are relevant for the disease. And as I told you, we have samples as early as stage 2.
Stage 2, okay. Thank you.
Just one question, easy. It should be easy. That is, what makes you better than all the competitors that are now working around the world in liquid biopsy with RNA? That I guess that is not something very exclusive. In fact, is one of the top shaped trends now in oncology. Huh?
Yes. So, so removing the stool-based analyses that are very specific of colorectal cancer, but there are many liquid biopsies coming up. Most of them are using DNA. The problem with DNA is that, first of all, they are only applicable for cancer or mutation-driven diseases, and normally they are not specific of one cancer, because the same mutation in different cells can cause different types of cancer, which means that, yes, they are good to detect your cancer, but still you don't know where the cancer is. While with RNA, since we expect the RNAs, or we are detecting RNAs that are being lysed from the cells that have cancer, and we not only can detect actually the mutations that come from exons, but
my point is not technical. It's in terms of what makes you different. Means if you have any kind of legal protection or patent or whatever about that.
In terms of the IP, at the moment we don't have any patent, and we are studying actually if it's the best way to patent or not this method. We can also keep it as a trade secret, because as soon as we patent, I think it will be very difficult to enforce patents, because we cannot really make sure what others are doing. They might be copying us. So what we are doing at the moment, or our first strategy, will be to have a server in the cloud where the data is uploaded and provides the result, but we don't need to disclose exactly which our markers are, which will happen if we patent. But that's why we would like to discuss with an expert on the field that could advise us.
Can you speculate on the price of each of the tests and how it could compare with the cost of other methods?
So the cost of one single analysis of RNA, it will be around 500 euros, which is still lower than the most expensive methods that are being offered at the moment, which are the DNA liquid biopsies. But the beauty of this method is that now I'm talking about colorectal cancer, but in parallel we are already starting a new cohort in lung cancer, and we want to try many other cohorts that then we can test at the same time with one single blood draw, and with the cost of just one test we can test for thousands of tests, so it will become more cost effective as we develop.
Thanks, João. So